A transparent amphipod moves across a trench floor beneath pressure capable of deforming poorly protected machinery. Nothing braces its body against a visible wall, and no rigid shell holds the ocean back. These depths, water presses from every direction with thousands of tons distributed across its surface. The mystery is why living tissue continues working, while human designs require thick metal pressure housings. If you carried an air-filled container downward, its shrinking volume would expose the crucial difference. Deep-sea animals avoid large compressible spaces, allowing internal fluids to remain near surrounding pressure. Equal pressure does not eliminate biological stress, but it prevents the simple crushing imagined at the surface. The real challenge occurs at molecular scales, where pressure alters proteins, membranes, and chemical reactions. Survival, therefore, depends less on brute armor than on maintaining flexible, functioning cellular machinery. Our investigation descends with pressure as the spine, following its effects from body cavities into individual molecules. Near the surface, gas-filled swim bladders help many fishes regulate buoyancy with modest pressure changes. As depth increases, gas compresses strongly, making such spaces difficult to maintain without active adjustment. Many deep-sea fishes reduce gas spaces, replace them with lipids, or possess no swim bladder. A snailfish hovering above the bottom uses a watery body and low-density tissues rather than a rigid pressure vessel. Its form seems delicate because resistance to compression is not the same challenge as resistance to impact. Water and bodily fluids compress only slightly, so tissues experience pressure throughout instead of across a hollow boundary. A rigid submersible keeps one atmosphere inside, forcing its hull to resist a vast pressure difference. An unarmored animal filled with water carries almost no comparable boundary across most of its body. This mainstream physical explanation accounts for gross survival, but it does not guarantee normal biochemistry. Pressure changes molecular volumes and interactions, disturbing the shapes that let proteins perform specific tasks. Enzymes must bind substrates, channels must open, and structural proteins must assemble despite altered energetic conditions. Deep-sea proteins often show adaptations that preserve useful flexibility under cold, high-pressure conditions. Too much rigidity can slow function, while too much flexibility can destabilize precise molecular structures. Evolution tunes this balance through amino-acid substitutions, expression levels, and supporting cellular compounds. The adaptations differ among lineages because descent into deep water occurred repeatedly rather than once. Membranes face a related problem as pressure and cold pack lipid molecules more tightly together. Cells respond by adjusting lipid composition, often increasing unsaturated fatty acids that help preserve fluidity. A functioning membrane maintains transport, signaling, and energy production. A rigid membrane compromises all three. The animal senses conditions through cellular regulation, changes lipid synthesis, and protects membrane performance. Long-term evolutionary changes establish the available toolkit, while short-term acclimation can adjust proportions within limits. Those limits help explain why many species occupy restricted depth ranges, despite appearing physically uncrushable. Temperature, oxygen, food, and pressure change together, making pressure effects difficult to isolate in wild distributions. Laboratory pressure chambers separate variables, but capturing and maintaining deep animals can itself alter physiology. Researchers therefore combine controlled experiments with chemical analysis, genomic comparisons, and observations at depth. Each method answers a different question, and none alone recreates an entire lifetime under pressure. One molecular protector repeatedly associated with depth is trimethylamine N-oxide, commonly called TMAO. TMAO can stabilize proteins against pressure-induced disruption in many marine organisms. Concentrations generally rise with habitat depth among numerous fishes and some invertebrates. This pattern supports a protective role, while its exact effects depend on proteins, species, and cellular context. Imagine you following a fish deeper as its chemistry bears an increasing cost. High TMAO concentrations affect osmotic balance, so cells must accommodate the dissolved compound and associated water movement. There may be an upper biochemical constraint where protective benefits no longer outweigh osmotic and physiological burdens. Observations suggest such constraints contribute to the absence of bony fishes from the deepest trench floors. They do not establish one universal depth limit because temperature and lineage-specific physiology also matter. The deepest confirmed fishes are snailfishes, family Liparidae, specialized for hadal environments. Below their known range, amphipods, holothurians, microbes, and other invertebrates continue occupying deeper water. Their success shows that pressure tolerance is built from multiple biochemical solutions rather than one compound. Protein structures in deep organisms can also incorporate changes that reduce harmful internal cavities. Pressure favors states occupying smaller volumes, so cavities and weak interactions can alter folding behavior. Some enzymes trade stability at surface pressure for effective performance within their native high-pressure environment. Brought upward, those same molecules may work differently, reminding us that adaptation is context-dependent. Pressure also challenges genetic material indirectly through replication, repair, and the proteins organizing cellular processes. Microbes from deep habitats display pressure-adapted metabolism, with some requiring high pressure for optimal growth. Such piezophiles provide direct evidence that pressure can become a preferred condition rather than merely tolerated stress. Not every organism collected deep is pressure-loving. Some survive a wide range, and others depend on high pressure. Correct classification requires growth or performance measurements across controlled pressure conditions. Reproduction creates another test because eggs, embryos, and larvae may tolerate different conditions from adults. Population cannot inhabit a trench permanently unless its full life cycle succeeds there. Pressure influences development through membranes, cytoskeletons, enzymes, and cellular division, yet evidence remains sparse for many species. Some deep-sea animals produce large yolky eggs and develop without migrating to surface waters. That strategy keeps vulnerable stages within stable conditions, but demands substantial maternal investment. Other lineages disperse across depth, showing that pressure tolerance can shift during development. Without complete life history observations, adult capture alone cannot prove permanent residency. Feeding behavior reveals how pressure-adapted bodies translate molecular survival into ecological action. A Haddle amphipod detects carrion odor, swims toward the plume, tears tissue, and stores energy between rare meals. Flexible joints and muscles function because their proteins and membranes remain effective under native conditions. The consequence is rapid recycling of material delivered from shallower ecosystems into trench food webs. Holothurians process sediment, ingest organic particles, and redistribute material across the seafloor. Microbes transform dissolved and particulate compounds, sustaining chemical cycles beneath immense hydrostatic load. Pressure does not provide their energy. It defines the conditions under which energy must be captured. Food scarcity, near freezing temperatures, and geographic isolation often constrain these communities alongside pressure. Calling pressure the only extreme, therefore, hides the combined environmental problem deep life actually solves. It also misleads exploration, because an animal surviving capture may still have suffered invisible biochemical damage. Decompression can expand gases, alter membranes, disrupt protein function, and change behavior before laboratory observation. Pressure-retaining samplers preserve native conditions from collection through examination for selected small organisms. They reveal physiological activity that conventional recovery might erase or distort. For you, the animal's softness may suggest vulnerability. Yet softness can minimize dangerous compressible boundaries. Human engineering increasingly borrows this logic by placing electronics in pressure-tolerant, oil-filled housings. Comparison is useful, although living repair and regulation remain unlike manufactured components. Understanding natural pressure adaptation may inform enzymes designed for high-pressure industrial processes. Its larger value lies in defining where life can function on Earth and potentially within subsurface oceans elsewhere. That astrobiological extension is a hypothesis about habitability, not evidence of extraterrestrial organisms. Ocean change could also shift deep communities through warming, oxygen loss, or altered food delivery. These pressures are ecological rather than hydrostatic, but they act on organisms already close to physiological limits. Future work must measure intact cellular function across realistic combinations of pressure, temperature, oxygen, and nutrition. Long-duration pressure aquaria could reveal acclimation, reproduction, and multi-generational limits rather than short survival alone. Pressure-retaining imaging may document feeding and development without the confounding shock of ascent. Proteomic studies can identify molecules that change, but experiments must test whether those changes cause tolerance. Membrane measurements across depth-adapted relatives could separate inherited adaptation from immediate environmental adjustment. Researchers need more data from embryos and juveniles, the stages that often define geographic limits. Comparative work should include invertebrates and microbes rather than treating fishes as universal models. Precise depth records also require calibrated instruments and direct environmental measurements at collection sites. No specimen should be assigned a pressure adaptation merely because it was recovered from deep water. It might have fallen, migrated briefly, or arrived damaged from a different habitat. Repeated observation of active populations provides stronger evidence than an isolated occurrence. Return to the amphipod, walking beneath the weight of the trench's entire water column. The surrounding pressure enters its fluids almost evenly, so there is no hollow body waiting to implode. What you call crushing pressure becomes a molecular environment requiring adjusted proteins, membranes, and osmotic chemistry. Those solutions carry costs, restrict depth ranges, and remain incompletely known across most deep-sea lineages. Extreme pressure does not spare deep life. Evolution makes life function while pressure acts everywhere.